I will be speaking about something called coupled Markov chain Monte Carlo. To give some context, I'm a PhD student in the statistics department at Harvard. I've been working so far in Markov chain Monte Carlo, and this idea of coupling Markov chains is something we have found to show promising performance in common MCMC methods, and I will introduce this all to you today. Okay, so to kind of outline some of the goals before we get started with the content of the talk, I have two main things I want to get out of today's session. First of all, I want to introduce coupled Markov chain Monte Carlo. And secondly, I'm not a computational biologist by any means. So I would really appreciate some feedback and maybe some data set suggestions from you all. And in particular, the things I'll be talking about correspond to two papers. The first paper is L-like couplings, which has been published as of last year. And the second paper is coming online on arXiv today, actually. So this is really very recent stuff. I should also highlight the professor I've been working with, Pierre at Harvard. This is part of his larger research program related to coupled Markov chain Monte Carlo. So Pierre has some other papers in this field too. Okay, so now we will get started with the outline of the talk. So many of you may be familiar with MCMC or Markov chain Monte Carlo, but I will regardless give a motivation behind why we care about these things. Then I'll talk about this L-like coupling or coupled Markov chain Monte Carlo algorithms. And finally, I'll talk about something which hopefully will be relevant to this community, which will be an application area in genome-wide association studies. So the motivation here is from a statistical point of view, we have a target probability distribution, pi. We wish to generate samples from this target distribution pi. Why would this be a useful thing to do? For example, this is very useful if we want to calculate complex integrals, which are analytically intractable. For example, if you have the integral of two things multiplied together, the function h multiplied by the density pi, you can estimate this as this expectation, where these random variables xi are samples we generate from pi. So that's one basic motivation. The second motivation is when we want to do Bayesian inference. So if we care about a posterior distribution, which arises from our scientific problem, we wish to visualize this posterior distribution. And in high dimensional settings, doing this can be difficult. So this is a very generic motivation behind Monte Carlo methods. Now I'll introduce what I think is the landscape of different sampling techniques you have within Monte Carlo. And I'll separate this into three categories, which by no means is perfect or exhaustive. So we have a probability distribution pi, and we wish to generate samples from pi. How can we go about doing this? So number one is perfect sampling, which would be the diamond standard. So you have a generic distribution pi. You ideally want to sample from it perfectly and get independent samples. This is difficult to do in general. Pi can be from a very high dimensional distribution. The second thing is something called exact sampling, or more precisely, asymptotically exact sampling. What this means is now we generate a sequence of distributions pi t, such that as t goes to infinity, pi t approaches our distribution pi. A popular example which falls within the second category is Markov chain Monte Carlo, or MCMC, which has been very commonly used within the Bayesian computation community. And the last category I want to also highlight is something I would call approximate sampling. So here we choose to sample from a target distribution, pi delta, which is, we believe to be close to pi. And pi delta is something which is easier to sample from. Examples of techniques which fall under this third category would be something such as approximate MCMC or variational inference. Any questions so far or any point in the talk, please interrupt. Okay, so I'm going to be focusing on Markov chain Monte Carlo, MCMC, and highlight a motivating question behind why we care about MCMC. So here is the general setup for Markov chain Monte Carlo. We have an initial state for our Markov chain, which is x0. For every time t, we go forward from xt minus 1 to xt according to a kernel. You can think of the kernel as something which randomly pushes your chain forward through time. So there is some noise, and it's not a deterministic path. And we want to choose our kernel in a clever way, such that as we run the chain for a larger number of iterations, the distribution of the chain goes to our target distribution pi. So we want to come up with a clever choice of kernel, such that as we push the chain forward, the chain goes in distribution to our target of interest pi. Now, this all sounds nice in theory, because t in theory can go to infinity and can get us to the target of interest. But in reality, we must stop the chain at some finite time, capital T. This capital T might depend on things like time constraints, might depend on budget constraints, might depend on how much memory you have in your computer. So for some finite time t, which is every single practical setting where you run MCMC, the motivating question behind my first project is how close is the distribution of the chain xt, which is denoted by pi t, to our target of interest pi.
So Niloy, just for the audience, the K in a finite space setting would be the transition matrix, right? But are you imagining that you might not have a finite state space, or?
That's a really good question. So exactly, in a finite state space setting, K would be the transition matrix. In the continuous setting, where the state space, say, is like the real line, then K could be something else. For example, a simple example would be, imagine K, as I've highlighted here, to be something like the normal distribution centered about xt minus 1. Then we would have a random walk where every time step you can go greater or equal or less than your current state with equal probability. Good point of clarification, Alex. Okay, so now I will talk about a concrete example of Markov chains. This is like a very well-known Markov chain algorithm, which has been commonly used. And this is one special example of K. So here we have two steps. We first of all have this initial state of the chain. We have x0. So chain has to start somewhere, which is something we choose. And then from every step onwards, there are two things going on. First of all, we have a proposal. For example, the proposal Q can depend on the previous state of the chain. So Q could be, again, from another normal distribution centered about xt. But then we have this accept reject step. And this accept reject step involves accepting this proposal x star with this probability I've written under this minimum expression. So with this probability, we accept this proposal, and with 1 minus this probability, we stay where we are. So then when we reject, we stay at the previous time step value. And now I'm going to just give one simple example. Imagine we have a target, which is a standard normal. So we want to come up with a chain which goes to the standard normal distribution. We choose, just for this stylized example, a proposal which is also normal. And here is an example of a chain. We choose our chains to start at 10. Every time we propose according to a normal distribution, where the center is the previous state of the chain, and we have a variance of 0.25. And then we accept or reject this proposal at every step. Again, this may seem abstract, so I'm going to now show a picture to show what's going on. Here is an example of a chain. So to start with, looking at this figure, I want to start on the left-hand side of the figure at time equals 0. You can see this one trajectory of the chain always starts at the value 10. And you can see maybe around time 50 onwards, it seems to be around 0, and it seems to stay at 0. So just looking at this one trajectory, qualitatively, we can see maybe around time 50 onwards, we seem to be around the normal distribution. Why can I say this? It's because I know what the normal distribution looks like. I know the normal distribution is centered about 0, and it has some noise around 0. So to me, it looks like from time 50 onwards, this trajectory seems to have reached the target distribution. Now, a natural question would be, everything I've said so far is specific to this trajectory. Maybe I have another trajectory which behaves very differently. So previously, I just showed one trajectory of a chain. Now, I have generated this plot by plotting multiple trajectories. When I have multiple trajectories for each time t, so for example, for time equal to 100, I have multiple values from which I can plot a histogram. So I can plot a histogram for every time t. And from this, I get this caterpillar of histograms. Why is this caterpillar useful for visualization? It's because this caterpillar tells us at every time t, what does the distribution of the chain look like? So you can see, for example, from time 100 onwards, our histograms all seem to be centered about 0. Here is 0. And they seem to look like a normal distribution because we know what a standard normal distribution looks like. However, from time 50 and before, you can see that we start at the value 10, and then slowly we drift towards 0. So if we were to run this chain only for 25 time steps, we would be somewhere here. This would not be the target distribution of interest. So if you were to do science and this was your distribution of interest, and you decided based on your heuristics, okay, 25 time steps is enough, you would be getting misleading conclusions. On the other hand, if you decided to run this chain for 2,000 time steps, perhaps that's an unnecessarily large number of time steps. You might be being wasteful with your computational constraints. So that's the motivation behind this question I've written in blue. Why? How close is the distribution pi t to the target distribution pi? If we choose a value of t that's too small, thinking we've reached pi, we would get inaccurate conclusions and inaccurate inference. If we choose a value of t that's excessively large, then we are running our computations for more time than we really need to, and we're being wasteful in terms of our computational budget. So this blue question motivates the first paper of this talk. So how close is the distribution pi t of a Markov chain at time t to the target distribution of the Markov chain pi? This is the NeurIPS paper from last year with Pierre and Paul. And here I'm going to talk about coupled Markov chains now. Any questions so far? Please interrupt. Okay, so our motivating question is...
Question.
Yep.
Is what you're going to talk about specific to Hastings chains, or is it just about all kind of Markov chains?
That's a great question. So the methods I'll be talking about apply to like a large class of Markov chains. I've used Hastings to highlight like a very popular example people use in practice.
Great.
just to build some intuition behind MCMC. But if you are familiar with Hastings, the techniques I'll talk about apply much more broadly too.
Cool, thanks.
Yep. Okay, so we care about figuring out how close is the distribution pi t to the distribution pi. To make this precise, we need a notion of distance between distributions. So I'm going to highlight one simple example. So how close is the distribution pi t to pi? One popular way to measure distance between distributions is this definition called a total variation distance. So let's see what this expression is telling us. This expression is telling us we have some test function h. This test function h is bounded. So at most, it can take a value -1. At most, on the other side, it can take a value +1. We work out the expectation of this function h applied under xt, which is distributed according to pi t, and x infinity, which is something which has the distribution pi. If xt distribution is very close to x infinity distribution, that's saying if pi t is very close to pi, then this expectation will be very close to 0. So we would get a low value for the t. And you may wonder, okay, why have I chosen my test functions to be bounded by 1? And why am I choosing supremum? One practical use of total variation distance is this will capture errors for histograms and credible intervals. So what do I mean by this? Imagine we are trying to do Bayesian inference. We have a very complex posterior we care about. And we think we have a really good Markov chain Monte Carlo method, which gets us to this posterior. If we know that the total variation distance of that method at time 1000 is a TV distance of 0.01 to the true distribution of the posterior, then we know that the histograms we plot using our Markov chain's value at time 1000 has an error of at most 0.01. Or if we were to do credible intervals using our Markov chain, we would know that the error between the Markov chain credible intervals and the true posterior credible intervals is at most 0.01. So this gives you a useful measurement of error, which might come from MCMC. I would also point out there are other distances which the paper also covers, but for simplicity, I'm going to focus on this. Mathematics theory aside, I think the big idea here is we need a way to measure distance between different probability distributions. Total variation distance is a popular measure of such distance, which captures errors on things like histograms and credible intervals that are of interest to scientists. Okay, so now I've set the problem up. So I've said, this is the question we want to answer. How close is my chain at time t to the target distribution? I've given you all a way to measure what I mean when I say how close, in that I've given a distance between probability distributions. Now I'm going to give you the final answer from the project before going into detail. So this is one of the final kind of results from the project. So on the left-hand side, I'm showing the caterpillar of histograms we had previously for this very simple example, which was the Metropolis Hastings chain starting at 10, targeting a standard normal distribution. On the right-hand side, I'm showing the error bounds or the total variation bounds I get from my LLAC couplings method. Let's understand what's going on here. So in this simple example, the left-hand side plot is very informative because we can visually see around time 100 onwards, we seem to be in the right region in terms of converging to the target distribution. does this match with the error bounds we get? So in this setting, you can actually work out the exact error, which is given by the thick dark line. The dashed lines show the upper bounds in this error we get using our method. Our method has this parameter L. The higher the value of L, the better your upper bounds, but the algorithms take longer to run, which is a technical detail. But the idea here being, in this simple setting, where you can actually see the truth in some sense, in that you can see your trace plots very well. You know what a standard normal distribution looks like. We seem to be able to capture the error very well. In reality, if you were doing MCMC, say on a large scale data set, you will not have access to this picture on the left-hand side. Imagine you were doing MCMC on 100,000 dimensions, which is something I'll come to very soon. Then you might have to plot 100,000 of these trace plots, which is not feasible. And even more than that, you have to plot potential covariance trace plots, and so on and so on. Whereas the right-hand side picture captures this error, capturing all these dependencies very well. So this is the motivation and the kind of end result of this LLACT couplings project. I'm going to briefly describe how we come up with these error bounds now for this simple example. Before this, do we have any questions at this stage? I would say I have not explained to you all what I mean by LLACT couplings, which is the topic of the next slide. Okay, here we go. So what do I mean by LLACT couplings? When I say couplings, I'm going to refer to a pair of chains. Now the pair of chains, first of all, the first chain, which I show in black, has a head start, has a lag. By lag, I mean kind of like a head start here of 150 steps. So I first of all run the first chain in black for 150 steps, or for L steps. After running the first chain for L steps, I then run both the chains together in a clever way such that they meet at some random time. So here, the random time is around 190. In general, I denote the random time as tau L. And let's try to get some intuition behind why I'm doing this as potential non-statisticians. So if two chains are going to the target very quickly, then they will end up in the same region very quickly. And if they end up in the same region very quickly, they can exactly meet very quickly. So the intuition I'm trying to get here is, if we have fast meeting times, it means the chains mix very quickly and they converge very quickly. That's the big idea here. So we try to simulate these pairs of chains, and we try to simulate them such that they meet quickly. And the faster they meet, the faster the chains will converge. This is the motivation behind the bound. One thing I should also highlight is that the chains are marginally with the same distribution. So distribution of xt is the same as the distribution of yt at all t. And you can kind of see that. If you look at the time difference of 150, and then you look at the yellow line's trajectory and compare it with the initial trajectory of the black line, you can see they marginally follow a similar pattern. Based on this setup, what we're going to do is we're going to simulate lots of pairs of these chains to get them to meet, and use that meeting time to get upper bounds on the total variation distance. So you can see here the equation on the bottom of this slide. I have this error, which I define as the total variation distance. And then I have this upper bound of the error in blue, where the upper bound has my meeting time. So the idea here being, I simulate these coupled Markov chains. I measure, oh, the first coupled chain meets at 190 times steps. The second chain meets at 250, and so on and so on. After running these many times, I can actually get upper bounds to this total variation distance. This is exactly how we generate the plot on the right-hand side. So we run lots of coupled chains to estimate the blue part of the inequality, and we plot this blue part of the inequality on the right-hand side. This is the, yep.
Yeah, this is Luca. I have two questions, actually. The first one is that the middle expression in this slide does depend on h, the function that you use for weighting the distance, but it seems that the last expression doesn't have this dependence on h. So
Okay.
so that's question number one. And question number two, maybe if you can specify what you exactly mean that the two chain meet exactly, because it seems that for a continuous space, that thing should be zero no matter what.
So those are good questions. I'll start with the first. If you look at the expression I have, which I use to define total variation distance, you have h inside the expectations. But outside of this, we have something I've written as supremum of all h. So what I mean by this is we have a class of functions, h, which are between -1 and 1. I define this distance to be the h which gives me the maximum error. So because I'm taking supremum or maximum over all bounded h's between -1 and 1, the overall distance does not depend on any particular test function h. That's a good point of clarification.
Can I interrupt before you answer the second question with another question? What if you made the h1 on the support of the initial distribution and -1 on the final, then wouldn't you get a value of 2? Or am I confused here? Should you get, why do you get 1 instead of 2? And maybe
okay.
Doing that wrong.
Oh, I see what you're saying. Okay, so there is a definition of TV where there is an extra half outside of all of this.
I was wondering if there was a half missing to make this the definition that's in the plot.
Yes, I think that's a very, very detailed observation. That's a very good point. So people write TV in both ways, but you're right. If I want to write it such that the TV is strictly between 1 and 0, as I show in the plot, you want to add a half in here. Yeah. Good clarification point as well. But I think the method with which you came up with this is exactly what's going on when we look at the supremum of all h's. You think, okay, I know my distribution of xt. I know my distribution of x. What's the worst h I can choose to maximize this distance? As you were thinking about, Alex, when you were thinking of this -1 +1 choice of h. And I think, Luca, for your second question, so let's go back to this example. So the second condition I wrote in L-lag couplings is we want the chains with this lag of L to meet exactly at time t. So this equality. And your question was, xt, yt minus L might be in some d-dimensional continuous space. Like here it's just on the real line, but it could be that the two chains are moving in like 100,000 dimensional plots. Can we actually get the two chains to exactly meet? That's an excellent question. And the answer is this actually can be done. One way to kind of get some intuition behind this is we actually consider some kind of like a rejection sampling technique. If you imagine 2 points are really close to each other, with some probability, you can get them to meet exactly. And the details of this are in the paper with Pierre, which I will not go into for now. But to answer your question, it can actually be done. It's one of the surprising innovations of coupled MCMC. Does that help, Yuka?
Yes, thank you.
Okay.
So is it related to the fact that the jumps are not independent between the two chains, or?
Exactly. So, for example, looking at this plot again, we run the first chain for 150 time steps. From 150 time onwards, we actually evolve the two chains according to a joint kernel. So we don't let the chains evolve independently, because if they did evolve independently, then there's no hope they would meet, even in this one-dimensional continuous real-line state space.
Yeah.
But if you can get them to move jointly, then you can actually get them to meet with positive probability at every step. Good point of clarification.
And there's one more question from the audience about the lowercase t at the end of the blue expression. Is that supposed to be a capital T, or is it intended to be?
Okay, this is intended to be a lowercase t. And this kind of shows you what's happening with the bound. So what we do to generate the plot on the right-hand side is we simulate lots of coupled chains to get lots of values of tau. Tau is random for every coupled chains. So we get lots of random values of tau. And you can see this expression inside the expectation decreases as t goes larger. So for different values of t, we get something which decreases as t gets larger. Using this, we plot this line. So given, for example, 100 values of tau, for every fixed value of t, we can estimate this expectation just by looking at the empirical average for 100 values of tau. But using the same 100 values of tau, but a different value of t, we get another point in our curve in the upper bound on the right-hand side. So the t's here change because this small t corresponds to, I see, okay, so this, I think this small t should be the same as this big T. That's a typo at my bit. Yeah.
Okay.
Yes, good point. Okay. Just
Can I ask
to
another question?
Yep.
Sorry. Yeah, I'm Xinqiang. Can you classify? You have two random variables in that expectation, and which distribution do you do that expectation on?
Okay, so you're looking at this.
Yes.
This expectation, sure. So the random variable xt is distributed according to pi t. So pi t is the tth step of the Markov chain's distribution. So for example, for t equal to 25, pi t in this particular example will have some distribution centered somewhere between 5 and 10 based on our histogram.
Oh, Niloy, sorry to interrupt. I think just to clarify the question, because I had the same one earlier, it looks like you need xt and x infinity jointly defined, but I guess you can split up the expectation because of linearity, and then you don't actually need xt and x infinity on the same probability space. Is that?
Exactly. So xt and x infinity, it doesn't matter if they depend on each other or not because of linearity of expectation.
Okay.
Yeah.
I see.
Does that clarify your question? So by linearity of expectation, you can write this difference as difference of separate expectations.
Okay, I see. So another question is, in order to estimate the last term, so do you have to run this Markov chain like multiple times?
Yes, very good point. So you have to run, every time you run this coupled setup. So you run one version of this coupled chain, and then you get one value of tau.
Okay.
And then you run it independently. Now, a framework which works well in this setting is if you have parallel computation resources. Imagine you have access to, say, 10 computers. Then you can say, oh, I want to run 10 coupled chains on each computer. So you need independent values of tau.
I see. Thanks.
Yep. Okay, these are all very good questions. So I'm happy to hear you're following the talk with interest. Now, so this gives you some taste of how we come up with these bounds. To give some background as to why we care about this and how this compares to existing result, just as a point of reference. In applied probability, people study this question, and they often get mathematical bounds which look like this. They get bounds, upper bounds, where you have some constant c, which we don't know, and then we have a decreasing function ft. Ft could be something like exponential of minus t. So we know, oh, the chain looks to be converging fast, but because we don't know this constant, we cannot apply these results from applied probability in practice. Because given a t value, we don't know what this constant actually equals. And there is some work in statistics, and there is also some work in MCMC convergence diagnostics, which some of you might be familiar with. For example, the R hat, which is used in things like STAN often. What we do is slightly different to this. I've put this slide here just for reference. Please ask me questions maybe later on if anyone's interested in the comparisons of these. But to get back to our motivating example, so here we have something very simple. You might be wondering, why do we even care about measuring the error of this particular Markov chain? I can see when this converges just by looking at the plot on the left-hand side. So the question we ask is, can this simple diagnostic we have developed here work in large-scale settings? Now, lots of challenges happen in large-scale settings. First of all, as Luca mentioned, can we get these high-dimensional chains to exactly meet to get our method to work? If we can, then we can generate the plots which look similar to the plot on the right-hand side. Now, in high-dimensional settings, trace plots like we have on the left-hand side are not very informative because of lots of reasons. First of all, we do not want to manually visualize thousands of trace plots. And secondly, the trace plots might not capture the full properties of the distribution. There might be lots of dependencies between different components in a high-dimensional setting. So trace plots do not necessarily capture the full convergence, whereas the right-hand side plots capture it in a nicer way. So now I'm going to talk about a genome-wide association study application. So I've hopefully given a taster of why we care about this problem, in the sense that we want Markov chains to converge, and if they converge, assess the rate of convergence quickly. And now we want to look at a practical example where we apply this technique. This is very recent and ongoing work, looking at high-dimensional regression with an application to genome-wide association studies. So imagine this is the setting we work with. We have a genome-wide association studies dataset of maize crops. So we have roughly 2,000 different lines of maize crop. For each line of maize crop, we have roughly 100,000 covariates, which are linked to SNPs in the genome. So what do we measure about these crops? We measure for each crop the number of days taken for silk to emerge in the crop, which is part of the maize life cycle. So you can think of this as kind of how well the crops are growing. So we have only two inputs. We have our response, which is number of days, and we have this as a vector of size 2000. and we have a design matrix. Important point to highlight here is that we're in the high dimensional setting because our number of covariates is much larger than the number of data points we have. So this is the setup we're working with, and we want to do Bayesian inference. First thing you might be wondering is, okay, if we were to do Markov chain Monte Carlo, this might be very expensive, and we would have no idea whether the Markov chain would converge. This is the question I'll be going on to now. So here is a setup which some of you might be familiar with, but I will describe it regardless. So we have data, we have a design matrix and a response Y. Our model is simple linear regression, and we choose something called the half-T shrinkage prior. Now let's think about what this means. I want to highlight two properties of this prior. So our beta components depend on three things. First of all, we scale by the noise, which is a standard thing to do. But then we have two extra components to our prior. We have this Kc, and then we have this eta. Kc corresponds to this global shrinkage parameter. So you could think of Kc as how sparse we think the problem is. Etas correspond to local shrinkage parameter. So when eta is a large value or a small value, it will tell us which of the beta j's are null signals or non-null signals. A popular prior which falls under this framework is the horseshoe prior, actually. So the T distribution has this degree of freedom. When we choose this degree of freedom to be 1, which is actually then equal to the Cauchy, the horseshoe prior falls under this class of priors. Now, to give some intuition behind why we care about these priors, I'm going to show you what the prior density looks like. Why half-T priors? So here, I'm showing the prior density of each beta j. Now, two important features to highlight about these priors, which are vital for good statistical performance. If you notice about 0, the density actually goes to infinity. So we have a pole at 0. Why is this something desirable from our priors? This is something desirable from our priors because if we have something which has infinite density about 0, it will aggressively shrink the nulls. So all the null signals from our data will be aggressively shrunk to 0. On the other hand, we have fat tails or polynomial tails, which means it will not shrink components which correspond to signals.
Do you get exact zeros with this prior, Niloy?
That's a great question. So you don't get exact zeros. There is another class of priors called spike slab priors, where you do get exact zeros. Actually, this idea of having a pole at 0 is motivated by these spike slab priors. So the idea there being, if you want exact zeros, sometimes the computation of the posteriors become extremely challenging. So what people wanted to do is think about, okay, can we come up with a continuous relaxation of these exact 0 priors? And they thought, okay, what's the closest thing to an exact 0 but continuous? Something which goes to infinity in density about 0. So it's motivated by these exact 0 priors, but we allow this prior to still be continuous for computation reasons. For example, think about comparing it with the Gaussian prior. I think earlier this semester, Tamara Broderick and a student from her group talked about using a Gaussian prior in this setting. So for example, in the Gaussian prior, you will have finite density about 0 because normal distributions have finite density everywhere. And secondly, you have exponential tails. You don't have fat tails. So here we are allowing both infinite density and fat tails, motivated by good statistical properties. So we have now two features of this prior, which are good for statistical estimation. But this presents challenges.
Niloy, that's a great explanation. Another reason why I asked the question, that there are cases without an atomic mass at 0, like the Laplace prior, right, from the lasso, where you still get exact zeros. So, but
okay, so
is that different?
That's a good point. So imagine we had the Laplace prior. Then you don't have infinite density about 0. But the maximum a priori estimate then corresponds to the lasso. So if you were to do Bayesian inference with the lasso, with the Laplace prior, and you were to plot the posteriors would still not give you exact zeros.
Oh, indeed. Yes. Thanks.
If you wanted to use the posterior modes, then that would correspond to the lasso.
Great. And I also wanted to give you a 5-minute time check, and I will stop interrupting you.
Okay, sounds good. Yeah, so we do not get exact zeros, but we will get values of beta for the nulls, which are arbitrarily close to 0, based on this infinite shrinkage, infinite density about 0 from the prior. Now, these two properties, fat tails and a pole at 0, are desirable because of statistical estimation. But these present big challenges. So you can imagine in this setting, we have this infinite density for a pole at 0. We have fat tails. We have a data set of dimension 100,000. Choosing to run this type of posterior distribution using generic methods is actually pretty challenging. You will notice that, for example, if you were to run popular methods like HMC, because you have this density going off to 0, going off to infinity about 0, you will run into overflow issues and numerical error very quickly. So, and secondly, the thing I want to highlight is even if you do have a good Markov chain algorithm, assessing convergence of this algorithm is very difficult. So this motivates the project. Now I'm going to give a sneak peek as to what we end up getting. So without going into too much details, we look at a simple Gibbs sampler for a half-T prize, and we use the LLAC couplings method I introduced in the first half of the talk to diagnose the error of these Gibbs samplers. And we find something which to us seems very surprising in a positive way. We find that our Gibbs samplers, based on our methods, where the state space is 100,000 dimensional, converges in roughly 750 steps. So we were very optimistic. We were very surprised by this because this gives you a reason to be optimistic about doing MCMC even in large scale settings, in that even if each time step takes time, you actually converge in a relatively small number of time steps. So your burn-in bias here is only roughly 750. Whereas people previously may have thought, okay, if my state space is 100,000 dimensional, maybe I need to run my chain with 10,000 burn-in or some kind of heuristic based on a large number. So the methods we look at seem to suggest that you can actually develop MCMC chains which converge even in this large setting in a relatively few number of steps. That's the highlight sneak peek about the second paper. And just to wrap things up, I just wanted to...
Oh, Niloy, we have another minute. Can I ask you a question on the last slide?
Yes.
So how expensive is each iteration? And if you want things like posterior means, then even after the burn-in, you still have to sort of take a bunch more samples, right? So, and is that still kind of tractable? Like, are you finding realistically that it is?
Yeah, very good questions. So to give an idea of the complexity of this chain per iteration, it's of order n squared p. So it's linear in dimensions, which is actually compared to the current Markov chain algorithms, that's the about as fast as you can have. So I would say if you have a large data set, especially in terms of n, it can get expensive quickly, but it's feasible. For example, this simulation, just looking at my numerical times, it would take maybe, I would run it, maybe it'll take like 1 day, which for GWAS is still pretty impressive. And the second question you asked about, oh, we just don't care about the burn-in. We also care about MCMC estimates. Say we care about posterior means. I would like to highlight, there is actually a paper which I have not worked on by Pierre Jacob, the professor who's involved in these projects, where he answers that question, showing how couplings can be used to get really efficient estimates of things like posterior means.
Great, that's great.
Can I ask another question? Is there time?
Yep.
Yeah. So for your upper bound, is there assumptions on the distribution? Imagine there are like multiple mode distribution, and the burning steps only, get stuck at one of the mode and never reach another mode.
Yeah.
So you will never know there's another mode. So how can you get a upper bound over there?
That's a really good question. So one thing you can kind of see is if you look at this upper bound plot, do you see around time step 500, there is a plateau. There seems to be this kind of plateauing effect in the upper bound around time step 500. Things like this is kind of related to this question you referred to. The upper bounds hold under assumptions, which for this GWAS dataset, we do verify. But one intuitive idea to think here is the more independent samples you run to generate this plot, the more reliable your bounds.
That's cool. So the y-axis is your is a proven upper bound, right? It's not the exact distance. It's an upper bound for the TV distance.
Yes. Okay. Yes. And also we generate this using Monte Carlo methods itself. So this line also has some error.
Okay. So it's a noisy estimate of a provable upper bound.
Yes, that's a really good way to put it. The reason I tried to present here today is to get you all interested about some of the performance and some of the empirical results we've been seeing, and just to hopefully get some interaction. I'm sure you have really interesting applications and GWAS datasets, which I know nothing about. So please reach out. The code for all of these is available online, and you can reach out to me over email. Thank you.